Okay, I can see people are starting to enter the virtual lecture hall. We will wait just 1 or 2 more minutes and
Sure.
let everybody get settled in, and then we'll get started. Looks like we're going to have a great turnout today.
Looks like.
We'll give it just 1 more minute, and then we'll get started. See a few more people entering. Okay. Looks like it's leveled off, so we'll go ahead and get started. Good afternoon, good evening, and good morning, everyone, depending on where you might be chiming in from today. I am Rob Burgess, and I head up business development for Sino-Biological. And I want to welcome all the attendees as well as our speaker to the next installment in our series of Lock and Key technology-based webinars. We have a fantastic webinar today from a great speaker focused on high-affinity antibody generation and new technologies related to that. Before I introduce the speaker, I just want to outline one housekeeping issue. And that is number one, that you type into the chat box, just greet us and say hello and tell us where you're from all over the world. We love to find out where individuals are calling in from and enjoying these webinars. And also, if you don't mind, if you have good questions for the speaker, just type them into the chat box and we'll withhold all the questions until the end of the lecture. And then I will walk through as many questions as I can verbally with our speaker. And so with that, it's my pleasure to introduce to everybody Dr. Dongxiao Feng. Dr. Feng is currently Vice President at GenoBioTX. Dr. Feng received his PhD from the Chinese Academy of Medical Sciences and completed postdoctoral fellowships both at Oxford University and the University of California, right here in San Francisco, where I'm calling in from today. Dr. Feng has 30 years of experience in monoclonal antibody development using hybridoma, phage, and yeast display methods and technologies. He also led the development of the second generation of HuMab mice at Bristol Myers Squibb, as well as the development of a fully human antibody transgenic mouse platform back in China. In addition to that, 2 antibody drugs were developed by Dr. Feng's team, and they have been approved in China and are at phase 3 clinical trials in the United States. He joined GenoBioTX 2 years ago as the Vice President of Business Development there. Dr. Feng, it's a real pleasure and honor to have you today as our guest lecturer. You may now share your screen and begin your lecture.
Okay. Can everyone see my screen now?
Yeah, it looks great. Thank you.
Okay. Okay. Thank you very much, Rob, for the introduction. And thank you, everyone, for attending this webinar. And I'm Dongxiao. It is really my pleasure to give you the presentation for the generation of high affinity and diversity therapeutic antibodies to self or high homology antigens. As you know, antibodies have been widely used for all kinds of areas, basic research, therapeutic, diagnosis, and so on and so forth. But there are still 3 major methods to generate monoclonal antibodies. The first one, as the most conventional one, is a hybridoma from immunized the rodent. The second one is a phage, whatever surface display, either phage, yeast, mammalian cell, bacteria. So you can set up an antibody library, so you can screen antibody from the library for monoclone. And also the most advanced technology is a single cell cloning. You can identify, characterize antigen-specific binding B cells from all kinds of animals, including human, to clone the antibody gene for sufficient recombinant expression. And these are the three major technologies for monoclonal antibody development. For therapeutic purpose, so human antibody is the most preferred antibody platform. So I'm going to give you some introduction of the fully human antibody transcriptional mouse platform we have right now. And as you know, this is the evolution of therapeutic antibodies. So at the beginning, people use mouse monoclonal antibodies, but then they found this very severe drug, anti-mouse drug reaction in human body, then it will take off the shelf very soon. Then people start to use chimeric, humanized, or fully human antibodies. The purpose to develop therapeutic antibodies is to reduce the immunogenicity of all kinds of antibodies. Right now, humanized or fully human antibody are the two major antibodies that have been approved in the market. But in the future, maybe artificial intelligence, AI, may be applied. But right now, it's still on the paper. Now, no AI antibody has been approved or in the clinical stage yet. So we are still focused on fully human antibody development. As you know, there are quite a few, a handful fully human antibody platform being developed. So from the company I worked for, the BMS, we developed the world first fully human antibody mouse model called HuMAb. And since then, and a lot of platforms have been developed. So we just developed a new fully human antibody development mouse model called SmokeMAb. So this is a strategy how we humanize the heavy chain of human antibody in the mouse. We select 24 VH genes of the most frequently used VH fragment in human and include all the D region and the G region and replace mouse endogenous VH, D, and G region in the mouse ES cells and keep the mouse constant region. This is the strategy we use for heavy chain humanization. And this is a strategy we use for kappa chain, the light chain humanization. So we select the 21 most frequently used variable region for kappa chain and keep the G region, all the G region of human, and we use the mouse constant kappa light chain constant region also. This is a strategy we use for the RG kappa humanization. Because in human, more than 95% of antibodies are from a kappa region. That's why we didn't humanize the lambda. We just fully knock out the lambda region of the mouse. So in case to avoid any mismatch between human heavy chain and the mouse kappa, mouse light chain. And we make this mouse, and we try to track the development of the mouse B cells in those SmokeMAb mice. As you know, if the B cell development is not good, so the spleen size will be much bigger. So this is we measured the size of the spleen from our SmokeMAb mice and compare it with the wild type C57 mice. As you can see here, the spleen size are almost the same, which means the B cell development is normal in this mouse model. And then we analyze the leukocyte cell subpopulation in the spleen and the peripheral blood. This is the gauge that we used to identify, to characterize the cells in the using FACS method. You can see here the mouse has normal T cell, B cell, and NK cells. All the BCR markers on the B cells are similar with that of the wild type mice in the spleen. This is the analysis in the blood of the SmokeMAb mice. So from here, you can see the smoke mice and the C57 mice have similar T cell, B cell, NK cells, and also the BCR marker are the same with each other. Then we immunize the mice with an antigen. So this is a antibody titer in the mouse blood after 3 times of immunization. From here, you can see that after third injection, the antibody titer in the mouse against the antigen reached to 1 to 100K, which similar to that of wild-type Barbicane mice. That means the human antibody has been successfully developed in the mouse. For the next step, we conducted single B cell sequence analysis, RNA-seq. So this is a sequence analysis of heavy chain variable region. From here, you can see those are all human heavy chain sequences. This is the light chain sequencing result. These are all human light chain sequences. And then we try to find out if all the variable region and D region or G region have been used in those mice. You can see that this is a pairing circle of human IgHV and IgKalpa V in the smoke map mice. This we test 2 antigens. You can see that all the variable region in heavy chain and light chain are being used, and they can paired to make a fully monoclonal antibody. It's quite similar to that of the pattern in human. So then we test the frequency of all the variable region and compare the frequency with that of a human and also K-map. As you know that K-map developed the first genomic chromosome replacement fully human antibody platform. We compared the frequency of our mice and with that of human and K-map. You can see that the frequency is very similar with among of the 3 data. This is for the G region comparison. You can see that similar to that of a K-map. Then, as you know that during the maturation of the B cells, a lot of mutations will be introduced into the CDR region, both CDR1, 2, 3, and to increase the affinity of the antibody to the antigen. From here, you can see both the light chain and the heavy chain have successfully been introduced a lot of mutations to for antigen binding. So that which means that the antibody can be successfully matured in the mouse B cell. The human antibody can be successfully matured in the mouse B cells. Then we try to develop hybridoma from those cells using the same antigen. And from here, you can see from one plate, there are quite a lot of positive clones in a 19-6-well plate. And then finally, we identified 5 positive hybridoma cell lines, which can secrete human antibody against the antigen we used, compared with that of a PBS negative control, which means that we have successfully developed the fully human antibody transgenic mouse platform for therapeutic antibody development. For therapeutic purpose, as you know that you want to a lot of antigen that we want to use to immunize the mice, they have very conservative reagents. Sometimes even in the mouse, human or rat have the homologous very high. But for the no matter it's a human or mouse, they have a mechanism to eliminate the antibodies against the self-antigens. This is the mechanism how our immune system can eliminate all the B cells that can recognize our self-antigens. All the premature B cells are induced to apoptosis before they can develop to secrete antibodies against our own antigen. That's why, and it's really hard to develop the antigens with a high homology or self-antigens in rodent. Right now, to overcome those hurdles, people have tried different methods to generate antibodies against the self or high homology antigens. Firstly, you may make a fusion protein with the antigen you want to use and with a high antigenicity T cell epitopes, so you can stimulate the immune system of the rodent. But for this method, you have to design the antigen very carefully, and also the purification and the characterization of the antibody is really complicated. Second way, we can use the SLE like autoimmune mouse models, for example, NZB W strain for immunization, because this mouse can develop the self-antibodies by itself. However, and the affinity of the antibody is quite low, and also it's a cross-reaction with other antigens. The possibility is very high, so it's not quite successfully so far. Alternatively, we can also use a knockout background mice to immunize for the immunization. So you can, because the antigen have been knocked out from the mouse, the antigen you are going to immunize for this mouse will be recognized as a foreign antigen. So the mouse can develop very high immune response. However, the lack of knockout animals for many targets of interest prohibits the routine application of this strategy. It is even worse for fully human antibody development, because for all the fully human antibody platforms developed by all the companies, including KMAB, no matter the Cytogen or Gene Pharmatech, they are all based on wild-type background. It's really hard to make knockout mouse from those fully human transgenic mice. At least it will take 1 year or even longer to get a knockout mouse model for antigen immunization. Most of our clients, they cannot wait for 1 or 2 years to get a mouse for immunization. So it's not very easy to use. This is the 2 example. People can develop self or high homology antigens in the background, in the knockout background mouse. This is the antibody development of a BCHE antigen for human antigen, and they knock out the BCHE of the mouse and immunize the mouse, the knockout mouse with the antigen. As a control group, they also immunize the wild-type mice. So for the knockout mice, the antibody titer can reach to 50,000 after 3 times of immunization. But for the wild-type mice, the antibody titer is 0 after even 4 times of both injection. So the monoclonal antibody developed from the knockout mice and can recognize both human and rat antigen. It's good for Western blot, for immunohistochemistry analysis. The antibody can also recognize mouse endogenous BCHE antigen. This is the Western blot analysis of the mouse antigen. So this is the very successful report for the monoclonal antibody development in the knockout background. This use a recombinant antigen for the immunization. We can also use DNA for the immunization. This is an example how people developed anti-prior protein antibodies in the prior protein knockout mouse model using DNA immunization. From the knockout background, people can get very high titer of the antibody titer in the knockout mouse, but not in the wild-type mice. So this is the FACS analysis of the antibody in the mouse get from the mice using the antigen positive cell line. You can see very significant peak shift in the knockout mouse background, but not in the wild-type mouse background, which means that you can use either recombinant protein, or you can use DNA to immunize those knockout mouse to make a monoclonal antibody against the high homology or self antigen. And but as you know that if you cannot get the knockout mice in a very short time, you have to wait maybe 6 months or 1 year. That's why GenoBioTX, we developed a new technology. We can get fully human antibody from the antigen knockout background in 3 months. So the benefit of this technology that in the of course, the 3 months fully 3 months homologous mice are F0 mice, but you can analyze the phenotypes of the phenotypes directly in the F0 mice. You can also validate your hypothesis earlier and adjust your research direction swiftly. And of course, you can initiate your highly affinity and diversity antibody development in 3 months. And because for immunization, of this method, we use CRISPR-Cas9 technology. For basic research, you may worry about the off-target effect. But for immunization, you don't really care about the off-target effect. You just need the gene knockout background for the immunization. So you can start your immunization process right away after 3 months. And for this, we have 2 examples how we generate the knockout mice. This is the generation of a CAR4 knockout mouse model in 3 months. And this is a strategy we use to knock out the CAR4 gene. From this result, you can see that from 7 new pups, we identified 4 homozygous knockout mice. And also 2 of them are heterozygous, 1 is still wild type. And for this technology, we can also make permutation mice. This is an example how we introduce a G2C mutation into the CPA gene in the mouse model. From a sequence result, you can see it's clear a homozygous for C and not a heterozygous or Y-type G. We can complete both knockout or permutation in as short as 3 months. So the mice are ready for research or for immunization. So for this technology, we can provide at least 3 to 5 homozygous small map mice per project. I think this number is good enough for antigen immunization, since normally people for monoclonal antibody development, we use 3 to 5 mice at the beginning. And we can provide we can guarantee the antigen gene is fully knocked out, and by PCR and sequence analysis data. And however, this technology has its limitation. For example, if the fully knockout of the gene can cause embryonic lethal or cause some prenatal death, we cannot use this method to generate homozygous F0 mice for immunization. But we may introduce some permutation to the targeted epitope. Maybe it's a alternative option. It also takes 3 months. Sometimes the functional region of the antigen is very conservative. You can introduce a permutation to cause new antigenicity to the antigen, but to keep the mice alive, then you can immunize the mice with Y-type antigen to simulate to get the antibody against the Y-type mice. So we can try both methods for antibody drug development. So based on those 2 technologies, the SMOC-map fully human antibody platform and our 3 months homozygous kill mice technology, we can make 1 mouse, 1 antigen. This is the fully human antibody mice tailored to your antigen, to your specific antibody development project. Since the antigen is fully knocked out, it's considered as a foreign antigen. So you can develop high affinity and high diversity antibodies against whatever epitope on the surface of the antigen. So this is a timeline for this project. So we can prepare the kill mice in 3 months. No matter it's a Y-type mice, if you want humanize the mouse monoclonal antibody in the future, or you want to develop the fully human antibody directly, we can prepare the mice in 3 months, and you can immunize the mice in 2 to 3 months, 4 times or 6 times injection. Then you can use all the 3 conventional methods to generate your hit. Normally it takes 1 to 3 months for hybridoma, for display, or for single cell sequence. Right now, since a lot of companies, they want to development bispecific or even multiple specific antibodies. So we also work with Immuno, a company based in Beijing, use their XFAB to develop the bispecific antibody for you. For this is the structure of the XFAB platform. They just, you can see here, you can fusion expression of the VH1 and the VH2 and conjugate the VH1 and VH2 with the CH1 as one chain, and the other chain is the VL1, CL, and VL2. And the two chains can be connected with the disulfide bonds. This is the basic structure of the XFAB. For this kind of structure, because this is the FAB structure, the antibody can keep its original affinity, so you don't need to optimize the structure if you don't use the SCFV structure or other form of a bispecific antibody. And you can also make multiple therapeutic antibody in this structure. The most important thing is that this antibody can be easily purified with a protein L resin, just like a protein A we used for monoclonal antibody for purification. In the true cells, the expression level of the XFAB bispecific antibody can reach normally 2 to 4 grams per liter. The highest may reach 6 grams per liter. And because it's the asymmetric double chain structure, the mismatch is pretty low, and you can use conventional method for purification and can lower your cost for the CMC process. And this structure has been successfully used for a lot of bispecific antibody molecules. This is some example and the expression level and the EC50 of the bispecific antibodies. And also 2 antibodies have been approved in China and in the US for clinical trials using the XFAB structure. This purification of the XFAB bispecific antibody that is expressed in 293 or 2K1 cells. This is SGS gel analysis of the purified antibody, the bispecific XFAB antibody. This is some single clone, choke clone expression level analysis. And this SEC, this HPLC analysis of the purity of the bispecific antibody. So by this combination of the technology, the SMOCMAP fully human antibody platform, the 3 months KO homologous mice technology, and XFAB, we can meet your requirement from the very beginning to the multiple specific antibody development project in 1 year. So I think that's all for my presentation today. Thank you very much for your attention. If you have any questions, yeah, I'm happy to answer your questions right now.
Thank you. Dr. Feng, that was a fantastic presentation. We really appreciate it. I would like to remind everybody to please type your questions into the chat box. We have plenty of time left for questions. While y'all are doing that, I had a couple of questions for you, Dr. Feng. The first one is, it's just a very general question, but you had mentioned that you have 6,000 mouse knockouts now, like a library of those from your potential customers for GenoBioTX to choose from. For antibody development. Like, how did you go about choosing those particular 6,000 targets? Like, what percentage are cell surface receptors? What percentage are maybe secreted proteins or enzymes? Like, why those 6,000?
Okay, sure. Since the company was established in 2000, we have almost 25 years experience. At the beginning, we want to set up a library for all the genes. We can knock out or conditional knockout of the genes. So during the past 24 years, we have accumulated all those 6,000 knockout mice. I think a lot of them are cell surface, almost all the cell surface antigens. We have the knockout mouse model in stock. So we just, but we couldn't keep them all alive. If you want some, if any client wants those mice, we can just rederive and ship the mice to the client in 3 months, or we can also help the client to develop the antibody in using those kill mice. Thank you.
Answer. Looks like you may have, there we go. You're back online. Thanks for that. That's a great answer. We've got a few questions from the folks, but I'll ask one more. It's a little bit unrelated to your talk today, but your bio says that you had developed a couple of antibody-based drugs in China. They were approved in China, and now they're in phase 3 clinical trials in the US. Can you tell us a little bit about those 2 drugs, those antibody-based drugs?
Oh yeah, I went back to China at 2010. At that time, a lot of Chinese pharmaceutical companies are working on biosimilar antibody drugs in China. And I joined a company called Luye Pharma in China, and I helped them set up all the antibody platform, develop the first fully human antibody transgenic mouse model for them. And then I also lead 2 biosimilar projects. One is Avastin, the other one is Prolia. So now those 2 drugs have been approved in China, and the Prolia one has been approved for FDA. Right now it's at phase 3 clinical trial. That's 2 drugs. And also the company has developed quite a few innovative antibody drugs using the fully human antibody transgenic mouse model, but still at a clinical stage. Since I left the company 5 years ago, I don't have too much update for those antibodies right now.
Great. It's very exciting work, though.
Thank you.
So we'll go to our audience then. The first question comes from Kong Chen. Kong is a graduate of Nanjing University and is now currently in Detroit. Kong is stating here, the literature reports that the N glycans of recombinant antibodies produced in CHO or 293 cells are different from those in human serum. Does this difference affect the affinity of those antibodies?
I don't think the glycosylation can affect the affinity, but it may cause some autogenesis in the antibody expressed in CHO or 293 cells compared with that from human serum. But right now, if you look at all no matter it's a humanized mouse monoclonal antibody or fully human antibody on the market, almost all the antibodies will cause anti-antibody drug response in the patients. So it's really not a big issue, but not as severe as some like the mouse monoclonal antibody. So people may mention that, but you really don't need to disclose those glycosylation. And so you can go ahead to use whatever technology you think is you prefer, no matter it's a mouse monoclonal antibody combination or you have some fully human antibody platform you can reach out, you can just use the fully human antibodies. You don't need to do any downstream homogenization, maturation, optimization. It's kind of easy to move on to the next stage.
Okay, great. And I think you may have answered this question, but when Kong is also asking about the patent filing process, either in China or the US. Is it necessary to disclose glycosylation information when filing like a monoclonal antibody patent?
Sure. Yeah, for innovative antibody drugs, you don't really need to disclose those information. You just disclose all the preclinical toxicity, whatever preclinical data to the FDA. But for biosimilar drugs, you cannot only keep the amino acid sequence similar to the original drug, you also keep the similar glycosylation pattern as the original drug. So that's why you have to disclose all the information to FDA.
For biosimilars,
yeah,
right?
for biosimilar drugs, yeah.
Got it. Yeah, because the purpose is for it to be similar. So
yeah,
really is the sequence supposed to be the same, but the 3D structure and the post-translational modification should be
of course, yeah, everything have to be the same, to be similar. Even you cannot see my drug is have a high affinity or higher efficacy than original drug. It's not good.
Right. Okay, well, we'll give our audience 1 minute or so more to ask some other good questions. I'll ask him one more since we're talking about glycosylation. It's my understanding that if you can glycosylate an antibody, it actually binds much tighter in certain instances. Have you ever done any work? Have you ever seen a necessity to use a glycosylase to remove glycan groups and improve affinity of an antibody?
Yeah, I see quite a few company working at some antibodies that without any glycosylation at all. I know that a company in Bay Area called Satchel. They use bacteria, the cell-free E. coli system to express the antibody. Everyone knows that E. coli has no glycosylation mechanism, so
Right.
all the antibody are glycosylation free. So it doesn't matter if we want to use true mammalian cell express antibody or E. coli express antibody. I think FDA really required is safety and efficacy.
Yeah, yeah, that makes sense. We actually have some knockout mammalian cell lines that are lacking some glycosylases.
Yes, yeah.
And as a result, the antibodies that we make from those tend to bind a lot tighter than other antibodies. So just since we were on that subject, I wanted to bring it up. Looks like we don't have any other questions from our audience, so I think we could probably close a few minutes early today, if that's all right. First of all, I'd like to thank all of our attendees. We had a pretty good turnout today. We appreciate your attendance and the good questions, Kang. We appreciate those. Dr. Feng, I also want to thank you for an excellent seminar. We appreciate you volunteering to do this. It's very exciting work that you've done, both before GenoBioTX and now with GenoBioTX. And so congrats to you and your team and to everybody at GenoBioTX. It's a fantastic company, got a great future ahead of you.
Thank you very much, Rob. Thank you everyone for taking your time to join our webinar.
Yeah, and one more person, I'd like to thank my good friend and colleague, Priscilla Hernandez, who executed this webinar from a technical perspective. She always aces it. So thank you for that, Priscilla. And I guess now we'll sign off and I'll bid everybody a good afternoon or good evening. Thank you.
Bye bye.